Hi everyone, we are going to start series on embryology. So embryology is the study of prenatal development of gametes, so-called gametogenesis, then fertilization, that is the formation of zygote, and then the development of embryos and the fetuses. So in these sessions, we'll try to cover the conceptual basis of almost every part of embryology, starting from the gametogenesis, that is the formation of gametes, which includes spermatogenesis in males and oogenesis in females. Then the fertilization. After fertilization, that is the formation of zygote. Later on, the cell division and cleavage of zygote, and then implantation in the uterus and placenta formation, followed by development of embryo in the first week, second week, and third week, from a cell mass into a two-layered embryo to a two-cell layered embryo to a three-cell layer embryo, trilaminar embryo, and then the tissue differentiation, and thus the formation of various organ systems like development of central nervous system, development of cardiovascular system, development of respiratory system, development of urogenital system, and development of gastrointestinal system, and other miscellaneous topics. We also try to touch upon the clinical points while we're discussing each and every systems. So stay tuned for a fresh video on this embryology series every day at 5 p.m. And so please do like, share, and subscribe to my channel. So if you can see over here, gametogenesis is nothing but formation of male gamete and female gamete. So for this formation of these gametes have to develop from some precursor cells, obviously. So what are they? These precursor cells from which the gametes are formed are called as primordial germ cells, PGCs, or otherwise called as primordial germ cells. Primordial germ cells. From where these primordial germ cells are coming from? So these primordial germ cells are derived from a special cells of the two-layer celled embryo called as epiblast cells. So if this is the embryonic disc, on the embryonic disc, the cells above the embryonic disc are called as epiblast cells. The cells below the embryonic disc are called as hypoblast cells. So epiblast cells, during the course, we will learn how this structure of embryo is formed from the zygote. We will learn during the course. But for now, just remember, these primordial germ cells are coming from epiblast cells. Not only these primordial germ cells, even most of the cells, most of the tissues, all the three germ layers like ectoderm, endoderm, mesoderm are also derived from these epiblast cells only. So epiblast cells are very, very important. How this is happening? See here. So epiblast cells are differentiated into primordial germ cells, and these primordial germ cells migrate through the primitive streak. This is the primitive streak at second week of embryonic development. Through the primitive streak, they travel, and they cross the primitive pit to enter the yolk sac. They enter the yolk sac endodermal wall. This is the endodermal wall. They enter the yolk sac endodermal wall at fourth week. From the yolk sac endodermal wall, they further migrate to the genital ridge area near the urogenital sinus. What is this genital ridge area? Genital ridge area is nothing but the intermediate mesoderm tissue which is located near the urogenital system is called as genital ridge. So this genital ridge is the place where the genital system, the male and female genital system, the testes and ovaries do develop from this region called as genital ridge. So they migrated sixth week near the intermediate mesoderm near the intermediate mesoderm at urogenital sinus called as genital ridge. So this is how migration of primordial germ cells are occurring from the epiblast cell layer towards the genital ridge at sixth week of development. So from sixth week of development, this primordial germ cell starts its function, that is starts undergoing gametogenesis, but in males and females, but in different fashion. How it's occurring, we'll see right now. So first, regarding the spermatogenesis is nothing but the formation of sperms, which is the male gametes. So how this is occurring and where this is occurring? This is obviously occurring in the male genital organ testis. In testis, if you know the basics, in testis there are some tubules called as seminiferous tubules. These are the seminiferous tubules in which these primordial germ cells are present. So what happens? These primordial germ cells undergo multiple mitotic divisions. These cells are called as spermatogonia or spermatogonial cells, which has the capacity to divide. So these spermatogonium cells have an ability to differentiate into a special type of cells called as primary spermatocyte. So these primordial germs are which are diploid in nature. These primordial germ cells which are diploid in nature, they differentiate into spermatogonium through mitosis, and these spermatogonial cells through mitosis again they differentiate into a type of cells called as primary spermatocytes. This is called as spermatocytogenesis. So formation of primary spermatocyte is called as spermatocytogenesis. That is formation of spermatocyte. Genesis is formation, as simple as that. So this primary spermatocyte is diploid in nature. First point. Next, this primary spermatocyte is the cell which undergoes meiotic division, first meiotic division to form secondary spermatocyte. After first meiotic division, obviously two cells are formed which are haploid each. So these haploid secondary spermatocytes now undergo meiosis 2 or second meiotic division to form 4 spermatids or 2 spermatids each. This formation of spermatids from secondary spermatocytes, which are also haploid over here, all 4 are haploid over here, is called as spermatogenesis or spermatogenesis. That is formation of spermatids. Next, these spermatids are immature forms of sperms and they undergo nourishment from the surrounding cells called as Sertoli cells in the seminiferous tubule. After nourishment, these spermatids finally are converted into the mature sperms with a head, tail and a cap. Head, neck, tail and a cap. This is the mature morphology of the spermatozoa or mature sperms. So this formation of spermatozoa or mature sperms from the spermatids is called as spermiogenesis. Very important. Formation of mature sperms or spermatozoa from the spermatids. From four are formed. This is called as spermiogenesis. So this is how spermatogenesis occurs in the male genitals. So some important points is each spermatogonium, which is formed from the primordial germ cell mitotic division, can undergo around 5 to 7 cycles of division to form primary spermatocytes. So, and one spermatogonium therefore gives rise to 64 to 512 spermatozoa, because every primary spermatocyte has an ability to produce 4 spermatids and 4 spermatozoa. Yeah. Regarding the oogenesis is peculiarly very important, and it has its own characteristic features. Oogenesis, where it is occurring? Oogenesis is occurring in ovary. Oogenesis is formation of ova or mature ova from in the ovary, yeah, from primordial germ cell called as oogonia. Yeah, so obviously these primordial germ cells, otherwise called as oogonia, undergo multiple mitotic division to produce many oogonia. One oogonia in the embryo itself, these oogonia in the embryo itself, they start dividing into primary oocyte. And now these primary oocytes are not further developed in the embryo. These primary oocytes are undergo first meiotic division. The first meiotic division starts in this primary oocyte, but they are arrested at prophase 1. You know, prophase, metaphase, anaphase, telophase. In this, the first phase of meiosis, that is prophase 1 of meiosis, they are arrested. In which stage of prophase 1? If prophase also has many other substages called as diplotin, pachytin, like that. So in diplotin phase, in exactly the diplotin phase of prophase 1, very important MCQ for entrance also, this in diplotin phase of prophase 1, this primary oocyte is arrested, which is also a diploid cell. This occurs before birth in the embryo itself. They are arrested like that. Until when? Until even after the birth, during the childhood, this ovary is inactive. Yeah, these oogonia cells are present in the follicles called as primordial follicle in the ovary. Yes. So in this primordial follicle, this oogonium is resting or oogonium is present, and they are arrested also. They are also arrested in this primordial follicle itself. So when this arrest is resumed or when they are released from the arrest, it's my question. So the answer for my question is they are released from their arrest immediately during puberty. What is the physiology of puberty? The pulsatile release of GnRH or the pulsatile release of gonadotropin releasing hormone. So due to pulsatile release, that is, the GnRH hormone is released in pulses. Suddenly a pulse of GnRH is released during puberty. That physiology is responsible for puberty, which we may be studying in physiology of puberty later on. But this pulsatile release of GnRH is resulting in puberty. At this puberty, whenever this puberty is happening, Immediately then, this prophase 1 which is arrested, diplotin of prophase 1, this oocyte which is arrested at diplotin phase of prophase 1 is released from its arrest and completes its first meiotic division. Yeah, it completes its first meiotic division, but to form one secondary oocyte and one polar body. One secondary oocyte and one polar body called as first polar body. This first polar body will degenerate and the secondary oocyte undergoes the second meiotic division in the growing follicle. So the primordial follicle is, during the course of development, they are converted to primary follicle, and whenever the puberty strikes, they are converted to growing follicles, and finally they form mature follicle. In the mature follicle, the secondary oocytes are there, but the secondary oocyte does not undergo second meiotic division right now, because the secondary oocyte, when it starts its second meiotic division, again it is arrested, again its cell division is arrested, but where at metaphase, at meta of meiosis 2 for 24 hours. This is very important. For 24 hours it is arrested at metaphase 2. Okay, so the second meiotic division is not completed right now. So next, what is the fate of this arrested secondary oocyte? Arrested secondary oocyte which is haploid, which is haploid cell, which is a haploid cell. So within these 24 hours, either this egg has to undergo ovulation and undergo fertilization. Yes, this is one fate. So if at all within these 24 hours it undergoes ovulation and fertilization, this cell is released from its arrest and it completes its second meiotic division. What happens if the fertilization does not occur within this metaphase arrest? If the fertilization does not occur within these 24 hours of metaphase arrest, this secondary oocyte will degenerate. This secondary oocyte will undergo degenerate. This secondary oocyte dies, or this secondary oocyte degenerates. That's how it happens. So that's what it's happening. So what's happening over here? The secondary oocyte is arrested in metaphase 2 and it is immediately ovulated, and fertilization has to happen to complete its meiosis 2. So once the fertilization is occurring, it results in zygote and a second polar body, and second polar body will degenerate. Yes. So we will see in the next session in a video, in an animated video. And after ovulation, that's what I told, ovulation is happening. After ovulation, they come into the ampulla part of the fallopian tube, which is a site for fertilization, and the fertilization is completed and the meiosis 2 is released from its arrest, that is released from its metaphase arrest, and thus the zygote is formed and the second polar body is formed. The second polar body dies and the zygote is responsible for the formation of the embryo. And what happens to this follicle in the ovary? So after ovulation, this mature follicle, after ovulation, it is converted into a yellowish scar called as corpus luteum, which has a function of producing progesterone, pregnancy hormone called as progesterone. Yeah. And yeah, so what happens if fertilization happens? If fertilization happens, what happens after metaphase arrest? Sorry, if fertilization happens, it completes its second meiotic division, and the corpus luteum keeps on producing the progesterone until the formation of placenta. So this will be the source of progesterone which maintains the pregnancy until placenta is formed, because later on placenta will be the major source of progesterone which is responsible for maintaining the pregnancy. Yeah. What happens if fertilization does not occur? This zygote is not formed, the metaphase arrested secondary oocyte will degenerate. Then what will happen? This corpus luteum will also degenerate to form corpus albicans if sperm is absent, which is a white color scar,